ARL Bio Pharma is a top contract laboratory partner for pharmacies, hospitals, and manufacturers. Sterility testing is essential to prevent patient harm due to microbial contamination. Our laboratory provides traditional sterility testing in accordance with USP 71, as well as validated alternative methods that comply with USP 1223 and are proven to be non-inferior to USP 71. Method suitability must be demonstrated for each drug product with correct sample quantities tested. ARL confirms method suitability for each sample prior to sterility testing. When a sample enters the receiving area for testing, our microbiologists carefully review each article, comparing the lot number, description, and sample quantities to the tests ordered. After confirmation, the microbiologist will also review any potential sample container breaches, such as loose caps, cracked containers, or improperly seated tops. After the sample passes review, the microbiologist will label the sample for testing and place it in a sealed, labeled container for transfer to the lab. Once in ARL's state-of-the-art HEPA-filtered sterility prep lab, the samples and all testing supplies are disinfected with sporicide and placed into pass-throughs adjoining the prep space and the ISO-classified sterility testing suite. The microbiologist then performs initial gowning procedures before entering the ISO 8 buffer room. The microbiologist will wash hands in a laboratory sink outside the sterility testing suite and don a hairnet, face mask, and shoe covers. The doors to the prep room and gown room are closed before opening the buffer room door. Once inside the buffer room, the microbiologist cleans their hands with a waterless, alcohol-based surgical scrub and dons a pair of nitrile gloves followed by a single-use cleanroom lab coat. The microbiologist will then enter the prep room and, following an additional disinfectant step of the materials, will place the materials in the pass-throughs adjoining the ISO 8 prep room and ISO 6 cleanroom. Our microbiologists use single-use gowning garb, which includes sterile gloves, integrated hood and face mask, coverall, boots, and goggles. The microbiologist disinfects the gown room table and bench with sporicide and sanitizes supplies with sterile 70% IPA. The materials are then aseptically opened, and the microbiologist will gown and pass the line of demarcation when finished. The microbiologist will use a mirror to inspect the gowning and make any minor adjustments necessary to ensure full coverage of all skin and clothing. After gowning is complete, the microbiologist will enter the ISO 6 airlock and wait for appropriate air changeover to occur. The microbiologist will then open the cleanroom door, step into the room, and allow the door to close. Once in the cleanroom, the microbiologist will prepare their laminar airflow hood for testing. Environmental monitoring media is set up and performed according to ARL's Microbiology Environmental Monitoring Program. All materials are disinfected with sporicide prior to entry into the laminar airflow hood. All critical points are sanitized with 70% IPA as materials are brought into the laminar airflow hood. The microbiologist then begins the sterility test. For membrane filtration testing, ARL utilizes closed membrane filtration units in combination with sterile rinse fluid. The microbiologist ensures each testing material is exposed only to first air during the execution of each sterility test and separates tested articles from non-tested articles following testing. During the testing shift, the microbiologist will perform additional personnel and environmental monitoring. At the conclusion of the shift, the microbiologist will also perform a proper negative control. The samples and testing items are then placed in pass-throughs adjoining the ISO 6 cleanroom and the ISO 8 prep room. The samples and testing items are then retrieved from the ISO 8 prep room and transferred out of the sterility testing suite. The microbiologist will then place the test containers into incubators, which hold temperatures at 30 to 35 degrees Celsius for FTG and 20 to 25 degrees Celsius for TSB. Following the duration of the validated incubation period, results are observed, reviewed, and then reported.